Okay, so case study from chapter 14. So it's a 94-year-old woman who is dehydrated. So she ends up in the ICU and eventually she develops pneumonia. So because of the pneumonia, they put her on strong antibiotics. And what happens is actually the strong antibiotics killed off her microbiome. So the microbiome are the organisms that should be found in her body, particularly her gut. And the thing that's left is one of the organisms that survives these kinds of situations is Clostridium difficile. Now, if you think about Clostridium difficile, Clostridium is actually a bacillus and it forms spores. Okay, so the spores are going to be important. And here's a picture. Okay, so what happens is the Clostridium, because it forms spores and the way that it lives in the intestines, it doesn't get killed by the antibiotic. So then it multiplies in the intestines and it produces these toxins A and B, and the toxins A and B cause really bad diarrhea, and they also form a pseudomembrane in the intestines. So half of the people that spend more than 4 weeks in the hospital end up with C. diff. So they call it C. diff. So one of the drugs that's used is and I spelled this wrong, nitazole, which it's a antibiotic that works on the gut, and the brand name is Flagyl. So sometimes you hear it called Flagyl, but it doesn't always work very well. So there's another drug called vancomycin, that's the brand name, and that works better than the generic vancomycin, but it's really super expensive. In the book she talks about, even with Medicare, it's like $2,000 a week for the treatment. Okay, so then lots of people relapse because it forms spores. So one of the treatments that they do to try to help with this is they'll do a fecal transplant. So they take feces from someone who lives in the same household as you, and they make sure that they don't have any disease, and they'll make a slurry and give you their feces, which has the healthy bacteria in it, and they'll do an enema. They also do an NG tube, so they'll insert the feces that way. Okay, there's a new drug out that's in clinical trials. It's called Difficid. So Difficid works against C. diff as well. So there's some articles online about C. diff and how bad it is. But it's a super infection, so it's taking advantage of the opportunity to cause disease. Okay, so history and definitions. So an antibiotic was defined by Waxman in the 1940s, and here you guys can read the definition, but he's defining it as a substance that's produced by microorganisms that have the capacity to inhibit the growth of other bacteria and destroy bacteria and other microorganisms. Okay, so they could inhibit, so they could be bacteriostatic, or they could be cidal. And the thing is, they're saying that they're coming from other organisms. So synthetic drugs are made in the lab, and then semi-synthetic drugs are drugs that are made from an organism. So you grow the fungus, like this picture is penicillin. So you grow the fungus, and then you extract the antibiotic from it, and then you might make modifications to it later. So history, they believe that the ancient Egyptians understood the theory behind antibiotics. So they put moldy bread in wounds. So it could be penicillin on the bread that they stuck in there. Now, in 1910, Paul Ehrlich, he kind of coined the term magic bullet. So he found this thing called salvarsan. And salvarsan they called chemotherapy. So they used chemicals to treat infection. So salvarsan was actually a mixture of a bunch of different chemicals. So it wasn't just one drug, but the active ingredient in it was actually really toxic. If you look here, the active ingredient is AS, and it's arsenic. So arsenic is bad. Okay, arsenic's toxic. So arsenic isn't ideal to use to kill bacteria. Okay, now Dalgak, I think that's how you say his name. He continues looking for antibiotics, and he finds that this dye, it's called pronicil, actually inhibits gram-negative or gram-positive bacteria. And then Fourneau figured out that the active ingredient in this red dye was sulfonamide. So sulfonamide is what sulfur drugs are. Okay, so sulfonamide was the active component. So they used to put sulfonamide packets in like the rations for the soldiers during war so that they could sprinkle it on wounds. One of the problems with sulfa drugs is a lot of people are allergic to them, and bacteria developed resistance to them really fast. Okay, so then the search continues, right? How are we going to treat? We need to find something to treat bacterial infections. So Fleming, he was actually working on Staph. So he's working on Staph aureus in the 1920s. And lots of microbiologists at that time, their plates would get contaminated. So here's Staph growing on his plate, okay, and you've got this fungus, which is the contaminant on the plate. And he noticed that there was a zone of inhibition where the bacteria didn't grow. So there was this zone, and we call it the zone of inhibition, where something was being produced by the fungus that inhibited the growth of the bacteria. So Fleming, other people probably saw this, but Fleming was the first one to realize the importance of it. So at that point in time, he was supposed to be working on Staph, so he wrote a paper about it and kind of had to put it away and do his job with the organism that he was supposed to be working on. But then in the 40s and during World War II, they were still looking for an antibiotic. So Chain and Fleury were actually chemists in England. So all this is going on in England. So they were chemists in England, and they start looking at Fleming's work. And they believe that they can actually synthesize penicillin in the lab. So they discover what the active ingredient is in this penicillium, and they call it penicillin, and then they try to make it. Well, they can't. They actually can't make it, so they have to grow the fungus and extract the chemical from the fungus. So it turns out that the Allies had a very good collaboration to develop penicillin. So England didn't have the infrastructure because it was destroyed during the war, so they didn't have the labs. So they worked with the United States and the USDA, and together they worked on the technology to develop penicillin. Okay, Waxman, he looked at streptomycin, and he started developing more antibiotics. Okay, now antimicrobial drug properties. So you want the antibiotic to be toxic to the pathogen, but not damage the host. Okay, so you want them to have a wide range between the therapeutic dosage level. Okay, so what damages the pathogen and what's bad for the person. Okay, so you can look at the difference between penicillin and arsenic. Okay, so penicillin, there's a very wide range between what's actually going to hurt people and what's going to kill the bacteria, whereas arsenic is toxic, right? So that's a narrow window. So arsenic's not a good antibiotic. Okay, now activity spectrum is what kinds of microbes does it affect. So we have narrow spectrum and broad spectrum. So you can kind of look, this chart is from the book, and you can see so some broad spectrum things, so like tetracyclines are on here. Some cephalosporins are broad, so they can kill gram-negative and gram-positive. Tetracycline pretty much works against everything except mycobacteria, which would be tuberculosis. So it's just like the window that what they can actually kill. The fluoroquinolones aren't on here, but they're broad too. Okay, so when do you use a broad spectrum? So broad spectrums really are kind of a shotgun approach. So it's when the patient's ill, you know that it's a bacterial infection, but you don't know what the organism is. So they just kind of hit it as hard as they can, as broadly as they can. Some of the disadvantages of using broad spectrum is that it destroys the microflora in the host, okay, so in the patient. And the more broad spectrums you use, the more antibiotic resistance occurs. Okay, now there's 5 things that I want you to know about how drugs work. Okay, and we're going to kind of hit on each of them. So one is they inhibit the synthesis of the cell walls. Okay, so they interfere with peptidoglycan formation. Okay, they can disrupt the plasma membrane. They inhibit protein synthesis. Okay, they act as anti-metabolites, and we'll talk about that. And then they can inhibit the synthesis of nucleic acids, so DNA, RNA. Okay, so the 5 ways that they work. So the first one is they can work on the cell wall. So they stop the formation of peptidoglycan. So if you can't make peptidoglycan, you don't have a cell wall. So, and this is kind of a stupid question, but what happens to the cells? The bacteria, the cells are going to die, right? So an example of those is penicillin. Okay, cephalosporin is another example. Okay, we don't have peptidoglycan, so in general they're not toxic to us. The active component of these chemicals are these beta-lactam rings, and I have a picture of those, but the beta-lactam rings are what interferes with the peptidoglycan. Okay, so they can work on gram-positive and gram-negative, but they don't usually work on archaea or fungi. Okay, now they can also interfere with membranes. Okay, so they kind of act as a detergent. So if you think about gram-negative, right, and the fact that they have a double membrane, okay, they can, the more membranes you have, if you're interfering with the membranes, they're going to mess you up more, okay? Bottom of it. So polymyxin is an example of this. So, oops, what? I guess I cut off. Okay, now, inhibition of protein synthesis. So if you remember from one of the first chapters we talked about, so bacteria have ribosomes, but they're different than our ribosomes. Okay, so if we can't make proteins, right, if we inhibit protein synthesis, the cells are going to die. Okay, so examples of those are chloramphenicol and erythromycin, erythromin. Okay, so they're broad spectrum, and they can have side effects. They can have side effects, but for the most part, they're going to act on the bacterial ribosome. Okay, now antibiotics that inhibit nucleic acid synthesis are things like rifampin or rifampicin. It's orange. Okay, so if you've ever seen it, it can actually make urine look orange, or if people take it long enough, it can make them look orange. So if we interfere with how DNA or RNA is made, then they're not going to be able to make proteins, and the cells are going to die. Okay, now anti-metabolites I think are really cool. So I always think of them, they're like posers maybe. So they're competitive, and we always talk about this, but this is a real example of it. So they're going to affect how the cell actually uses metabolites. So the example is sulfa. So bacteria, all cells need folic acid in order to go through mitosis, okay, in order to divide. Okay, so bacteria can make folic acid using this molecule called PABA. Okay, sulfa looks like PABA. So if you're going, you have this compound and you have this compound, okay, and you have to have PABA in here to make folic acid. What happens is sulfa resembles PABA enough that it interferes with the pathway. Okay, and then you can't make folic acid. And if you can't make folic acid, then the bacteria die. So it's kind of like a poser. It's pretending to be PABA, and by pretending to be PABA, it messes up this pathway. Okay, we don't have a PABA system. We get folic acid from our diet, so it's not going to cause any side effects with us. But bacteria are kind of smart, or they evolve quickly, so they have figured out ways to modify their PABA use. Okay, so there's other antimetabolites that mimic purines and pyrimidines. So they look like A's, T's, C's, and G's. Okay, so they get picked up, and then they'll stop the production of nucleic acids. Okay, now side effects. This is formatted weird because of importing it into this program, but side effects of antibiotics. So one thing is toxicity. Okay, so it can be harmful to cells. People can be allergic. They could have an immune response to the drugs. They can disrupt the normal microflora, so you end up with a super infection like a yeast infection or C. diff. Okay, or the bacteria develop resistance. So resistance can occur a couple different ways. So one could be a physical resistance where they're like hiding in the tissue, or there can be genetic resistance where we're adding a plasmid or we have a mutation occurring.